Good morning, everybody. Welcome to the Bridge webinar series. I am Laura Dubé. I'm the James McGill Chair of Consumer and Lifestyle Psychology and Marketing at the Desautels Faculty of Management. But I'm also the founding chair and scientific director of the McGill Center for the Convergence of Health and Economics, a center hosted in the Faculty of Management, but that has members from more than 7 faculty within McGill, and then from the same discipline around the world. We have created this center for addressing in a more impactful way the challenge that are of both health and economic nature that are at the interface of many systems across society, and at the middle of this, individuals themselves. This series, which we have been started over a year now, is between our center, the MCCHE, the Global Obesity Center at Johns Hopkins, and the Supercomputer Center at Carnegie Mellon. We are pleased in this new series to connect two new networks, the Mission Midas and the HBHL that is anchored here at McGill. So I will shift to Jay, who is leading this first network, and to Sean, who will be introducing our presenter of today. Jay?
Okay, thank you very much, Laurette. So I see a number of people from the Midas network have joined the call, but for those of you who aren't familiar with Midas, it is a collaborative network of research scientists who use computational, statistical, and mathematical models to understand infectious disease dynamics. So I'm the director of the Public Health Applications Group at the Pittsburgh Supercomputing Center at Carnegie Mellon, and we've co-sponsored the Bridge webinar series for some time under the leadership of our former director, Sean Brown, whom you'll hear from in a second. But the Midas Mission Group is the networking and education arm of the Informatics Services Group at Midas. And we at the PSC are Tom Maiden, who is also on the call, run this education and outreach. So I'm excited to kick off this Midas Mission co-sponsorship of the Bridge webinar series with Bill Wheaton, especially due to the long-standing collaboration between Midas and Bill at RTI, and the integral role that the synthetic populations that he's produced have played in the agent-based modeling work that I and others in the Midas network have done over the years. And so I'm going to turn it over to Sean, but I'd like to point out that the Mission Group is not limited to Midas network participants, and we will follow up with an email that will invite you to join Mission and to participate in not only these Bridge webinar series, but other networking and outreach events that we coordinate through Midas Mission. So with that, I'll turn it over to Sean so that he can introduce Bill, and we can hear a little bit about Sean's work too.
Okay, great, thanks, Jay. So my name is Sean Brown. Many of you know me already, but I am now the Chief Software Architect of the C-Brain project at the Montreal Neurologic Institute and Hospital at McGill University in Montreal, Quebec. And I'm also a part of one of our new co-sponsoring organizations, Healthy Brains for Healthy Lives, which is a large, about $100 million initiative here that was awarded to McGill University, headed by Alan Evans and Doina Prekup, who are both professors here at McGill University, with the goal of bringing infrastructure and neurological sciences to a number of different fields, diverse fields, including childhood development and diseases, brain disorders and dementia, and also behavioral science. And so my role here is to bring our infrastructure and technology to new areas such as agent-based modeling and other types of behavioral modeling, which is why we are interested in the Bridge webinar and very interested in our speaker today. I have the absolute pleasure of introducing Bill Wheaton, who I've had the pleasure of working with for what has it been, about 10 years now, Bill?
Probably so,
and
yeah.
Doing, yeah. So I started working with Bill right as I was cutting my teeth in agent-based modeling of public health systems. Bill was instrumental in creating the synthetic populations that Phil Cooley and myself used for modeling influenza diseases during H1N1, along with Bruce Lee from Johns Hopkins University, and have been working with Bill ever since on using his wonderful GIS technologies that he's been developing at RTI for creating agent-based modeling. So Bill is the senior geospatial scientist at RTI's Geospatial and Technology program at RTI International. He has more than 30 years of experience applying geographic information science and technology in environmental and social sciences, and he leads and coordinates the GIS tasks in support of diverse research throughout RTI and for external government and commercial clients. In addition to maintaining technical proficiency with GIS and related technologies, Bill has an extensive background in GIS and related topics to industry. He has worked in social policy, infectious disease modeling, surface water quality, groundwater quality, human and ecological risk assessments, air quality, survey research, and environmental contamination. I have gotten to know Bill as part of our work together in Midas, where Bill has been was one of the leads on the information technology resource for Midas, providing the synthetic population technology, which one could certainly argue has become the premier data set of its type around the world to be able to do agent-based modeling and simulation, and has indirectly, I would say, saved many lives across the world because of the work that people have done with his research. So I will turn it over. And one thing else I mentioned, Bill has his Master's of Liberal Studies from Duke University and a BA in Geography from Dartmouth College. So without further ado, Bill, I pass the baton over to you, and thank you very much for giving us a presentation.
Okay, thanks, Sean. Thanks for that nice introduction. Nobody ever said I might be responsible for saving lives. That's a new one. So, appreciate that.
Sure.
So let me start by just saying that I'm probably going to be more talking about synthetic populations here than agent-based models, even though ABMs are in the title. And secondly, I want everybody to look at this map here and see who can tell me where this is. What is this a map of? Any guesses? All right, well, I'll spill the beans. So this is the Los Angeles metropolitan area. So on the lower left is the Pacific Ocean. You can see the coastline running from the left down to the lower right. Pacific Palisades is the kind of blob that's jutting out into the coast there. And the blank areas in the map here, places where people do not live. So up in the upper left, I don't know, can you see my cursor as it moves around here on the slide? Okay, so up here in the upper left here, this is the Malibu Mountains, very sparsely populated. This is Malibu here. We go down into central LA, south central LA. The beach communities, this is LAX right here. These blank places are where people don't live, and the places where we have colored things, which are actually composed of individual dots. They look like blobs at this scale, but they're individual household dots. And can anybody guess what the color coding means? You've got yellow shading into bright blue. What variable might that be? All right, I'm going to tell you that's household income. Blues are high, yellows are low. And so you can see up in Malibu, you got high, Pacific Palisades, it's high. Some of the edge communities near foothills and mountains are high. Central Los Angeles is low. And so this is an example of the synthetic population that if you were to map this particular variable. And it provides a lot of advantages over typical census data because a census map of a administrative area is going to be color-coded all one color for an entire polygon. And this provides much more spatial resolution on the data. And to the extent where this is a very common place in the United States, Los Angeles, but when you are only looking at population and only looking at a realistic placement of people, it almost becomes an abstract kind of visualization of that place, but it's more realistic than the typical demographic map you might see. Okay, so moving on. So just want to acknowledge, as Midas was mentioned already, and this is really the source, the original source of funding for the work I'm going to be talking about, although later in the presentation I'll be talking about things we've done since we were involved in Midas. But Midas really was how we got going with the synthetic populations and provided the funding and certainly the application for which it would be useful, which is the infectious disease modeling. And I also wanted to highlight prior research that was instrumental in what we've done, and that is work done many years ago at Los Alamos National Lab in the development of the Transins simulation. And Dick Beckman there, who's the author of this paper, really laid out the methodology of using the statistical techniques to generate the synthetic populations in the way we do. There are other ways of doing it, which we probably don't have time to explore here, but this method that we're using is generally acknowledged as the kind of the primary method of doing it. So why do we need synthetic populations? What is their purpose? I like this quote from a paper done in 1999, and I'll just read it really quickly here. So microsimulation methodologies aim at building large-scale data sets on the attributes of individuals or households and at analyzing policy impacts on these micro units through simulation of economic, demographic, and social processes. If we do not have a micro database on individuals and households, then we have to simulate one. And this is really why we have to create synthetic populations, because we don't have a nationwide data set from the Census Bureau with everybody in it. You can't get that. It's protected. So we have to do our best using public data to create a representation of people and households that is pretty close to reality without having the protected information. So the primary purpose in creating them for Midas was, of course, for a source of agents, or people and households in this case, for simulations of infectious disease. But there are many other purposes for them, which I'll go into a bit later. So one of the key things about the synthetic population that as we generate it is that we account for household relationships. So we know for a household which people occupy that household. So we have household characteristics and we have the people who live in a household. And this is very important in a lot of kinds of models, even influenza, where transmission occurs through personal contact. And we know from experience that if a child has flu, caught it from school or wherever, then it's more likely that other people in that household will catch it as well because they're in close contact. And so those household-person relations are maintained in the data explicitly. So these are realistic, de-identified households and persons, and we can add additional characteristics to them from other sources, and I'll talk about that a little bit later. And the way we generate them is they are explicitly spatial. So looking at that map of Los Angeles, we place households based on population density, and we place the dots, not only so they represent the spatial distribution, but the characteristics as well. That's why the yellow areas that are low income reflect the actual low income areas, and the blues reflect the high income areas. So I consider these data to be infrastructure. A lot of people, when I talk about synthetic populations, they say, so what kind of model is it? And I always have to say, well, it's not a model, it's data. So it is simply a spatial database of households and persons in a country. And the reason we generated a whole complete U.S. version was because in the past, modelers, when they were trying to build a microsimulation or an AVM, they would have to scratch their heads and try to figure out how to create a data set to model. And a lot of times they would just kind of generate them for their own little study area, and maybe they were kind of abstract with just kind of age and gender characteristics. So we thought by developing a nationwide data set that was just there on the shelf for use would kind of open up the ability of people to develop models without having to worry about generating their own synthetic populations. So agent-based models was the reason Midas was interested. And agent-based models simulate the interactions of individuals and households and the environment. So we're not modeling like a group of people like you would with a cohort model. We're modeling individuals and households. So a person might have flu, and that person lives in a household, and there's some transmission dynamic from one person to another person. So each person has to be simulated, and characteristics of the disease, in the Midas case, influence who gets infected. So, so I just have a movie here I'm going to show that just is an example of the output of one of these infectious disease models. This is Pennsylvania, and the yellow dots are not actually individuals, because there would just be too many to map. These are actually % of people who are infected with flu over time at a block group level, which is a small census geographic level. And so over the course of an epidemic, I'm going to play it again here, you can see through the simulation where the influenza transmission is occurring, firstly in the eastern part of the state, and then it moves across to the western part of the state. And so the simulations really try to look at different mitigation scenarios, for example, to see what effect things like vaccination or closure of schools or other things might affect a disease epidemic curve. Okay, so an example of how the synthetic populations that we developed are used is in the FRED model that was developed at UPitt with some early support from RTI. Isn't that right, Sean? It was kind of Phil's early work that you guys were able to expand on.
Yeah.
Yep.
Mm-hmm.
And so what FRED does, which is really cool, is first of all, it's a web-based interface to a detailed model, and secondly, you can select any place in the country to run it on. So this is a perfect marriage, really, of a nationwide synthetic population data set and a model that can ingest that data into its dynamic simulation. And so when you run FRED, you can select the county you want to run it on. You can specify variables for the disease, for example, the transmissibility or the R0, and it will run on a machine at the UPitt or the Supercomputer Center, and it will create a map and a data set that shows you the results of that model. So very powerful use of these technologies. Okay, so how do we generate them, and what do they look like as we try to figure out what they should look like geographically? So first of all, in the U.S., we just the basics of some census geography. So counties, which are the heavy gray lines here. This is Durham County in the middle of this map here. where RTI is located, is divided up into census tracts, which are these, these yellowish lines, and then census tracts are further subdivided into block groups, which are these black lines. And for these block groups, we can get sociodemographic variables from what's called the American Community Survey in the U.S. So we get a lot of very good data for each of these block groups on, age distribution, employment, household size, whether you use, whether you're a renter or owner. All those kinds of census variables are available for these small geographic areas. And then we also have to think about another kind of geography, which are called public use microdata areas, which are in red here. And these areas are ones for which we can get in the U.S. a sample of the actual survey responses at a microdata level. So in this red area, we get 5% of the responses that are de-identified. And so we have microdata of each household and each person in each household, 5% of them in this area. So that provides the actual households and persons that are used to create the complete 100% synthetic population. And these are located all over the country. So when we're creating a synthetic population for southern Durham County here, the households and persons that we are going to draw from are from southern Durham County. They're not from California. They're not from Texas. They're from this particular part of southern Durham County. So that's an advantage of this way of doing it. And so within each block group, which are the black polygons here, we know the number of households, a very basic measure of population. And so we're going to create a record for each household that is assigned to each block group. So we're going to fill each of these block groups with the right number of households, and we're also going to characterize each household in a way that matches the census aggregated data for that block group. And when we're done, we'll have something that looks kind of like this. This is the dots, and this is a very old graphic where we simply randomly located each dot in the block group. Now, as you saw with that LA map, we've done a much better job of locating the dots. But you can see here that block groups that have lower density have fewer dots. Block groups that have higher density, they may have the same number of dots as a big polygon, but it looks blacker because they're denser. It's a smaller area. Okay, the way we decide which households to put into a block group are based on 4 characteristics, and we have some options for these. We can do some customization, but these are the 4 generic ones. So household size, age of the head of householder, the race of the head of householder, and the household income. So in a particular block group, like this, my mouse will show up here, this particular block group here, which is fairly low density, but anyways, what I'm talking about, what we're going to do is we're going to generate households, a list of households here, where when we're done, the set of households, and it looks like from the number there's about 300 or 400 households, that set of households will contain the right number of households by household size, age of head of householder, race of head of householder, and household income to match what the census says for that block group. As I said, we can use other attributes, tenure, age of the building, type of fuel, things like that, but these 4 seem to be probably the most useful in the widest number of applications. So we're matching on households, not on persons. So if you look at the distribution of age by person within a particular block group, it's going to be quite close, but it won't be as close as the age of the head of householder matching is. In terms of the data sets, what does it actually look like? There's really 2 key tables that we generate. One is a household table, and there's 1 record for each household, and the second table is a persons table, 1 record for each person, and they're linked together by a common ID. So this ID 9903 for a household. All the persons who occupy that have that same ID. And I've just highlighted one dot on the map here, this blue one. So we have a record for that household, which also has its latitude and longitude on it, and then the records for the person. So we have a lot of information about the persons, the age, gender. We do school assignments and work assignments, so we can decide where they go to school, if they're a school-aged child, or where they work. Earlier synthetic populations we generated did a better job of these schools and work IDs. Because of changes in data, we can't do as good a job as we used to on those. Okay, and then the key thing that I was mentioning about the distribution of the dots, this is a typical map of census geography. These are block groups color-coded by population. So blue is high population, yellow is low. And this is your typical census map, which is nice, but it doesn't tell the whole story because you have to assume when you see a map like this that population is evenly distributed within each geographic unit, and that is not the case at all. And so what we do is we take a second data set, a population density grid, and I've listed 3 here that might be used for this purpose, to do a better job of placing the households within these polygons. So these kind of brightly lit up areas are the places within this landscape where people actually live. So there's some towns in this area. There's actually a golf course community right here. That's what that is, towns here. And these other areas that don't have these little highlights are just, there's no people there. It's agriculture, forest, whatever. But if you just look at this map, it looks like people live everywhere, but they don't. They only live in certain places. So zooming in, here's a particular area that shows a block group here that has a fairly high population, but they're all the way over here on the eastern edge of this block group. So when we place our synthetic households, we have all the households for this block group, but we don't place them, oops, okay, there we go. We don't place synthetic households out here because people don't live there. We place them where the population density is. So that improves the spatial heterogeneity. If you're doing things like calculating distance to a medical facility, this is going to be much more precise than just randomly placing those dots. And here's an example of some even further improvements we can make to the placement of the households themselves. So on the left is the generic placement. Those yellow dots are the synthetic households based on this gridded data set. And so, and underneath that are blue buildings. These are building footprints in North Carolina. And so we don't really even care if those dots fall on buildings or not in most cases, because we don't want to get into any privacy issues or even have people think, oh, you've got a dot on my house, so you must know who I am. We don't even want to go there for the most part. So the density of the dots in yellow, this is zoomed in probably further than you would ever use the synthetic population here, but it reflects the overall density of population. On the right side, what we've done here is we've taken those yellow dots and we have placed them into the buildings. So it's called completion in GIS. And we have data on each building, which indicates the number of floors, and of course we have the size of the building just based on the footprint. And so we start placing the yellow dots and assigning them to buildings, and as a result we can color code these buildings now based on the number of households in them. And these light blue ones over here on the side, those are actually single family households, so they have very few synthetic households. These larger buildings here that are dark blue, we've placed a lot of households there, 10 to 25 or 25 or more. And this was done for the North Carolina Department of Emergency Management because they are really concerned about flooding and hurricane damage, because we do get hit down here by those things. And so they wanted to have a more accurate representation of where people live. And so this was some work we did for them. We can't do this nationwide because we don't have the building footprints nationwide. North Carolina did have them. Okay, so you might ask how accurate is the synthetic population? And so this table's kind of busy, but let me just kind of explain maybe one cell in the table. So we'll look at this one right here. This is household income, and this is, so bin 1 would be income less than $10,000. So we're trying to figure out how many households to put in a block group where the income is less than $10,000. And we've done that for the whole country. There's over 209,000 block groups. Only 36 of those block groups were cases where, let's say the American community says there ought to be 30 households with with income less than $10,000, and the margin of error is 5. So if we placed 50 households with income less than $10,000, we'd be outside the margin of error. But overall, across the entire 209,000 block groups, these are the count of block groups where we were not within the margin of error in the synthetic population. So very, very good here in terms of the accuracy. Another way to look at it is spatially. And so this, for North Carolina, shows the difference between the synthetic population counts and what the American community counts are for a particular variable. So this might be 3-person households. So there's 256 block groups where we got the exact correct number of 3-person households in a block group, and then in some cases we created more synthetic households than were expected. In some cases we created fewer. But overall, most of the block groups are plus or minus 3%. These are either light orange or light blue to what is expected. A few are outside that. There are only 3 that were greater than 6% and 9 greater than 6% on either side of the plus or minus. And when you move up to a different level of geography, these are now census tracts. They're bigger, and we get even better matching, and we go to county, even better matching. So it's pretty accurate for the most part. Okay, so let me move on to something else you want to do with synthetic populations, and that is add new variables to them. Because we're using census data for the generic synthetic population, what do we get out of census data? We got sociodemographics, age, gender, race, ethnicity, educational attainment, language spoken, whether you're in the military service, what kind of house you live in. But you don't get anything about behaviors or things like health. So what we want to be able to do is we want to imbue these people in the synthetic population with additional characteristics. So we call this enrichment. And so if we do that, we can then start to make maps and do cluster analysis and things like that by adding additional variables. And so this map here is a map of smoking risk. So it is the synthetic population, and what we did is we took a survey, in the U.S. it's called the BRFSS, Behavioral Risk Factor something survey. And one of the questions they ask is about smoking, tobacco use. And so we take that survey, which has demographics associated with it, age, gender, race, sex, ethnicity. And so we can predict, based on what kind of characteristics you have, whether you might be a smoker or not, and then we can map that. And so if the particular characteristic, like smoking, has a socio-demographic component, meaning certain kinds of people are more likely to do it, then we see a highly heterogeneous map of, in this case, smoking. We've also done it with the same process with food insecurity. So this map here shows in the reddish zones places where people are more highly food insecure. This is based on the what's called the NHANES survey, the National Household National Health. I can't remember. Anyway, it's a survey where they in the U.S. where they measure food intake, they measure height and weight, they measure all kinds of things like that. And one of the things they measure is food insecurity, which is really defined as your ability to feed yourself and your family well. And so if you're highly food insecure, it means you may not have enough money to make it through the month, or you might use a food kitchen, or you may not have healthy foods available. And so this allows us to map food insecurity at a highly detailed level, so that if we're doing an intervention or we want to try to build new resources into the community, we know where to place them. And then this is one where we tried to do celiac disease. So where is it likely that people have celiac disease? Because there are some sociodemographic correlations there. And this was done as a test for a pharma company. The black dots are gastroenterologists, and the blue are areas where there's a less likelihood of having celiac cases, and the reds are areas where there's more likelihood. And their interest was in enrolling patients for a clinical study. And so they wanted to know which GI doctors were located in places where there's a higher likelihood of people having celiac disease. And then finally, for this enrichment idea, I wanted to cover one that we did nationwide, which is assigning a body mass index to every adult in the synthetic population. And we submitted this to something called the NIH Obesity Data Challenge a couple of years ago, and it won second place, so we're very happy about that. There's a $10,000 prize, which was great, and would have been great if they'd given it to us, but went to the organization. But that's just kind of a joke. No one expected to get the money ourselves. Anyway, so the idea here is that in that NHANES survey, they have body mass index, and we know the characteristics of the people. And so we built a distribution, which is shown here in these charts. So this is for 20 to 29-year-olds, females, Mexican or Hispanic origin, and less than a bachelor's degree. And this is the distribution of BMI for that group of people. And so a BMI of 25 means you're overweight. A BMI of 30 or greater means you're obese. And so we take the shape of this distribution. We find all the people in the synthetic population that are 20 to 29, female, Hispanic, and have less than a bachelor's, and we roll the dice on this distribution and assign a BMI from it. So we don't assign just 30 or just 25. It might be 32.5 or it might be 27.6. But the result matches this distribution. And on the right-hand side is for a different sociodemographic group. This is 30 to 39-year-olds, male, Asian, non-Hispanic, and have a bachelor's degree. And it's a little difficult to tell because the scale is different. But this group of people generally has there are fewer people in this group with overweight or who are obese. So the BMIs that we attach to these people are going to be in the 20 to 25 area generally. So once we do that, we can then map the results. So in this map, because we can't show every person, first of all, people in a single household would sit on top of each other, and secondly, it's just too much data to show. This is actually mapped at a 250-meter grid cell size, so there's actually little boxes throughout here. And we simply summarize the people by the box they fall in, and this shows the percent of the population that is obese by these grid cells. And so we can see some clear clusters here. And the nice thing about this is that it's more detailed than what you can see from any U.S. federal government source. So if you look at CDC, they'll provide a map for you of obesity, and it'll just show counties. Let's say this county is 35.6% of the people are obese. Well, if you're doing a neighborhood intervention, that doesn't really help you get down to the level you want to get to. So this is just the percent obese map. This is a map of clusters. So this shows in the red areas, we're 99% sure that the distribution there is not the result of chance. It's a true cluster. So the reds are 99% sure that that is a high obesity area, and the blue is 99% sure that it's a lower obesity. It doesn't mean there's no obesity, but it's less. And then this is the Z-score. Okay, so these whole series of slides on enrichment are useful for not just modeling, but also for other kinds of applications like interventions in the field. If I wanted to create a study looking at a case control study and that's on obesity, well, I want my case area and my control area to have similar characteristics in terms of obesity. If I pick one area for my control that has very little obesity and an area for the cases that has high obesity, well, I'm not comparing like places. So it's another use for this kind of map. So one other thing that we've done and used a fair amount, not only in the U.S. but internationally, is to be able to age a synthetic population into the future. If you just take our 2010 synthetic population, it's static. It's not changing at all. If I'm running a flu simulation, seasonal flu simulation, well, the flu season only lasts part of the year, and so I don't really care if people are getting a year older or have a birthday within a 6-month period. It doesn't really affect the flu simulation. But for other kinds of purposes, it is important to be able to project into the future. And these would be things like the effects of climate change, which happen over decades, or if you're looking at obesity or stunting. If you can improve, let's say, the low birth weight births, I mean, this is a lot of work we've done in Indonesia, then that results in fewer stunted children, and that results in much lower healthcare costs and higher health for people who don't have that resulting problem. So we want to look at decadal-long simulations that reflect not only the fact that we gave some intervention to reduce low birth weight, but we want to see what happens to those people over time, over a long time. And so that's what this tool called FPOP does. It's a microsimulation that ages and projects the population in the future. And so this population pyramid just shows in the darker color maybe the current population pyramid, and then the outer one shows what might be simulated 20 or 30 years into the future. So it does change. So the FPOP model takes fertility, mortality, household formation, and dissolution, and migration. And for every person in the synthetic population, if they're a woman and they're a certain age and race and ethnicity, there's a fertility probability associated with them. And so at each year or time step of the model decides whether they've given birth in that year or not. Same with mortality. We have mortality tables that say what's the probability of somebody dying next year based on their age and race, ethnicity, and things like that. And so some people die next year. Some people get married, and some people come in or move out. And so it is a microsimulation because we're looking at a person-level basis. So that's the concept. And the idea here is that, well, let's say we want to simulate what the effect would be of a reduction in non-Hispanic Black mortality over time. So we tried 2% reduction, a 3% reduction in mortality for this racial ethnic group, and then we can play that over time. This is from 2011 to 2050, 40-year period, and we can see the count of persons under these different scenarios. So the default, where we use the default probabilities, is the red line. If we reduce mortality by 2%, we get this line, and by 3%, we get this top line. You can see that in earlier years, there's very little difference, very little effect, but over time, the effects get greater and greater because fewer people are dying because of this adjustment in mortality, then more and more people are born to those people who didn't die, and so on. So it's a dynamic simulation. Okay, and the last thing I want to show is another use of the synthetic populations having to do with resource allocation. And this is some work we did for LA County. This is one of the things we did for Bruce, Sean. And the idea here is that every county in the U.S. has to have a plan for mass vaccination. So if there's a smallpox outbreak or there's a novel infection for which there's a vaccine, these counties, every county will open mass vaccination center. They're called pods, pointed dispensing, where people can go and get the prophylaxis, whatever it may be. Well, in Los Angeles County, there's 205 of these sites they've set up, and those are the orange dots across the county. And they don't have enough staff to place into all these places. They need public health, professionals or nurses to give injections and provide vaccinations. And so what we wanted to do was help them understand which of these 205 should they open in the case of some sort of an emergency, to open the ones that are most efficient in terms of time of travel and distance. So where are the ones where you reduce the total travel time and total travel distance for everybody who needs to go to them? Now, clearly not everybody in LA, there's I think 6 or 8 million people, can just go to 7 of these facilities. But you can pick any number of facilities you want and run the simulation. But what this is doing is running a genetic algorithm, because the problem is just too enormous to even calculate here. We've got over 3 million households in LA, and for each one of these combinations of 7 out of the 205, we have to calculate the time and distance. On the road network, which is how we do this, it's not just as the crow flies. We actually do a road network analysis, and then sum up to get the total travel time, total travel distance. So we use a genetic algorithm for this problem, where we're simply trying out different combinations of 7 and calculating solving the problem, and then trying another 7 with a genetic algorithm that decides to swap a few out here and there. And eventually we come to a solution, which may not be the ultimately best solution, because that's the way genetic algorithms work, but it's probably a darn good solution. And it's something that can be done, within our lifetime, whereas I calculated it would be like trillions and trillions of calculations if we had to do this in a traditional method. So I'm going to play this as a animation here, and what you're going to see is about 600 different runs, where in each run we calculate the total time of travel and total distance to a different set of 7 points. And in the lower left here, this shows I think this is the total, let's call it total time of travel. So really bad solutions are up here in the white colors, and as we get better and better solutions, it comes down the rainbow down to blues and purples and eventually blacks. And so as I run this, you'll see a little white dot over here, which is the value of the solution, and then you can see it's selecting different red dots every time we run through a particular version of the genetic algorithm. So as it runs, it's getting better and better. So it's getting into the blue areas, which are already way down here on the scale from best to worst. And as it runs, it'll just eventually start to key in on certain red pointed dispensing locations that seem to really be key ones. And you'll start to see it slow down a little bit and which ones it's picking. But it's trying different combinations there's different genetic algorithm concepts like mutation and swapping and things like that, which is what it's doing here. So it's getting better and better as it goes along. And once it gets down into this purple area on the chart here, those are all pretty darn good. So the difference in these, in the total travel time may not be significant, but you can see that there's a lot of different ones that actually might be pretty similar in the value of the solution. But eventually, when it gets down to the end here, you're going to see that up here in Palmdale, this is part of LA County in the high desert. It's isolated from the rest of LA, but you see it gets highlighted and it stays highlighted. So in order to have the best solution, you really have to open that one up there, because there's people who live so far away from that and are isolated from the rest of LA County. You really want that one. And these others that have been selected here at the end are deemed the best solution here. So this is just one other application of synthetic populations to do this kind of problem. And I'm not showing the actual synthetic population households. You remember the map that I showed at the very beginning, all of those things are, there's 3 million households are underneath this and are providing the source of the distance and time calculations. Okay, so just to wrap up, so they're really valuable infrastructure for microsimulations and ABMs, but they also have many other uses. They can be enriched with additional behaviors and characteristics, and we use them for the resource allocation. We use them for risk assessment in terms of exposure to chemicals in the atmosphere or the environment, and so many other uses for them as well. And I'll close there, and if there are any, I guess we're going to have a question and answer period. One of the things I didn't mention is international work, so if anybody wants to know about that, can talk about that, and Canada specific, if that's something of interest.
Great, thanks, Bill. That was a wonderful talk. It's good to see all the great work that you're doing with the synthetic populations. Again, having worked with you over the years, it's great to see some of these things start coming to fruition. So well done. Thank you for a great presentation. So I'll open the floor to questions, and of course I will defer the first set of questions to Laurette.
Thank you, Sean. Excellent presentation, Bill. Thank you very much. A couple of general questions in the sense that you have this individual level map that is so rich in terms of information, and to what extent can this micro level representation, what are both the analytical interface as well as the mapping with the other level, like what impact, for instance, and I will be specific on food and nutrition, what impact individual behavior is much of a function of what different actors in whether it is health or whether it is food retailing, food manufacturing, that are doing and operating on different geographical and temporal scales. So how can we think about building upon what is currently done to improve our ability to map the various influences that operate on different scales, different geography to influence ultimately all those individual behavior? So that's my first question. And the second will be, it is very interesting that you have now, it seems, those synthetic population across the whole world, and I'm kind of just wondering what you see as the potential of using this as effectively as possible as we look at the local, the state, the regional, and all the various layers of influence, kind of taking a real one-world approach in examining some of those problems.
Okay, so for the first question, I think what you're asking there is how can we use this kind of generic data set to understand better what are the what are the influences on health from different aspects of a system, really, of what's out there in the world. And so I would say one of the things we can do is take some of these data that we have, let's say, well, just take something simple like physical access to health care. Okay, so distance or time of travel, we can look at even if a synthetic household owns a car or not, that's in there. And so we could simply use it to calculate variables such as a difficulty or accessibility variable from each household to a place of health care. And so just by doing that geospatial analysis, we can generate a new variable which then can be used in a model or further analysis, because that new variable indicates something about, let's say, the correlation between access to health care and location of households, the sociodemographic of households, and the distribution of health facilities. So I think in some cases, when you're developing a model, let's say the influenza models for Midas, we didn't need to add anything to the synthetic population, because all the models needed to know was, well, what's the R0 for transmission and some of the other natural characteristics of the disease, and then we just needed to know where people might happen to be in contact. But other models, we need other parameters that we don't know yet, and so I think this idea I'm just throwing out there is access to healthcare is a variable we can construct and then use that in a model that might attempt to predict what would happen if we added additional health care or if we provided free health care or whatever. So I'm not sure if that's getting to your question or not, but that is.
Yeah, it is. I would like to push it a bit, though, in the sense that in the context of eating or obesity or diet related, you will need to have many layers of influence in understanding the impact. So how flexible it is in being able to account with more than one factor at a time, type of.
Okay, so that would be dependent on the simulation model code itself. So with a microsimulation or an ABM, you can have many different variables that are being manipulated in the model. So I would say that's more a function of the modeling code than what's in the data itself. And then in terms of international, we have created synthetic populations for several countries around the world, and the ability to do that is dependent on the availability of census data mainly. And in some cases, we can build ones that are quite rich in attributes. In other cases, it's very basic, like we simply have the household location and number of people by age and gender. And so we can't really do it everywhere, I wouldn't say, at this point. That would be kind of a holy grail to do a global one, although some people have done very basic global synthetic populations, just creating a record for every person and placing them. But in the case of some work we've done in Indonesia on stunting and obesity, we can do a pretty good model at a province level. Okay, so in Indonesia, I think there's 35 or so provinces, and so we can see the heterogeneity from one province to the other in terms of providing, let's say, a micronutrient to pregnant mothers to improve birth outcomes. And because at a province level you have differences in the sociodemographics and things like sanitation and other factors like that, you can see differences in the effect of the micronutrient in different parts of the country. So that gets to your second question.
Yeah, thank you. If I may just, Sean, and I will stop here after. In your Indonesia study, and I was looking at your paper as well, the actual life course prediction about the consequences of stunting, to what extent is the current approach accounting? Is it expanding over time as a function of what happened if you are able to address undernutrition, or do you also account for the various layers of exposure that a person will accumulate over time? For instance, in undernutrition, you get exposed to obesogenic environment and you are more susceptible than if you had no. So this notion of looking at layers of environmental exposure is a question that I meant to push your thinking a bit. Yeah.
Right. No, I think that's a good question, and the modeling, the initial modeling we did in Indonesia would not have looked at. It just wasn't. We didn't have enough funding and time to build out those sorts of try to answer some of those questions. So the basic modeling we did, basically looked at, over the life course for a stunted person, what were the probable likelihoods of various, morbidities that would track along with a person who was stunted, without looking at other effects that might be happening to them, whether it's poor nutrition throughout their life or whatever it might be. So we didn't do it in that case, but there was a second set of modeling that we did where we were looking at sanitation, which could be considered an environmental variable in a way, but again, it didn't get to the dynamics of that over time. And we really just looked at, well, if a pregnant woman was in a household that had indoor plumbing and toilets, presumably that would mean lower amounts of I'm not sure what the epi term is, but lower gastrointestinal issues, which affect birth outcomes. So if the sanitation is better birth outcomes. And so in the model we did there, we said, well, what if we provided every household with decent sanitation, thereby reducing the issues with maternal health and improving outcomes. But just to summarize, I'd say we have not yet done a lot to look at changes in the environment over time, or changes in behavior over time, aside from kind of a trajectory of BMI and things like that, as is currently known.
Okay, thank you, Sean or Jay. I'm shifting back to you.
Sure. Yeah. Thanks. Thank you, Bill. Thanks, Lorette. I'll open the floor to other questions. I think Armin here from the MNI has a question for you, Bill.
Hello.
Hello.
My question should be fairly simple. I was just wondering, you were talking about, or answering one of the questions you were asked, that you might be able to essentially create new variables out of the existing data with the synthetic population. I was wondering, that's really cool that we can do that, but is there a way to verify before making a decision on that value?
So if we're using a survey as a source of an additional behavior or characteristic, then the statistical model that says, let's say for BMI, so we can look at the survey we have, and we can say, okay, these variables predict a BMI distribution of whatever. And so that's just the data, that's the statistics that show that for these sets of people, this is the distribution. So if the survey, if there's margin of error in the survey or other complications in the survey data, you might have higher or lower confidence in that distribution that you generate. So that's one issue. The second issue is if we are doing, let's say, a geospatial calculation to estimate distance to a healthcare clinic or hospital or something like that. There's obviously error in the street databases and error in geocoding and things like that. So, but at a regional level, we think it's pretty good. Now, for something like BMI, if we look at the BMI map that we produced, how do we know how good that is? I think that's really the crux of your question. And I would say the only way to know that would be to do a data collection task and actually do a survey to verify it. But I would say that would be very time-consuming and very expensive to do. And I think that if we have confidence in the synthetic population and we have confidence in the survey, the unhinged survey, which is very well regarded, then we do have a certain amount of confidence in the results, even if we can't exactly quantify it by a survey.
Thanks so much.
Yeah, one point there, Bill. One of our previous webinar speakers, and I believe we have that this talk is recorded and available on the YouTube. Are you familiar with Adam Drunowski from the University of Washington, Seattle?
No.
He was looking at alternative methods for making the similar maps to the one that you did for the BMI and comparing to your results for New York City, I believe it was. He was using different proxy indicators to see if he could match that. And so it might be, you might want to go back to our YouTube talk and see that, or look up his work.
Okay.
Your maps and looking at that exact question of verification. I think he had done a very big cohort study in Seattle and was using that to compare the data. Okay.
So he was comparing to the data that we had produced specifically?
The maps you produced.
Okay, cool. Yeah, no, I hadn't seen that. I'll go look at that. One thing to note that might be kind of related to this discussion is when we look at obesity, Hispanic origin is one of the highly correlated variables that predicts obesity. And so when we use Hispanic origin as one of the predictive variables, we are not getting to the point where we are evaluating, let's say, Hispanics from Mexico and Central America versus Hispanics from, say, Puerto Rico. And so when we simply predict Hispanic origin as a predictive variable, well, we may be missing some very big differences between those two Hispanic populations. So in LA, where the Hispanic population is generally from Mexico and Central America, you might really actually have different prediction of obesity there than you do in New York, where at least traditionally the Hispanic population is more than from Puerto Rico or other parts of the Caribbean. So that's something we don't know. So at least in the analysis we've done, where we're treating all Hispanics equally in their predictive power for obesity, and there might be differences there.
And that's I think that's another point to Armin's question that you just elucidated too, is that there is, one must be careful about the geographic extent that one makes these sort of extrapolations onto the population to make sure that your cohort is representative, and what other, the greater the geographic area that you try to apply the same methodology, you may be skirting over some heterogeneity that may be important to the metric that you're looking at.
That's right.
So, but yeah, no, it's still it's beautiful work. So I'm glad Arvin asked that question.
Yeah, no, it's important to know the limitations and some of the uncertainties for sure.
Cool. Okay, great. Are there any other questions from the audience for Bill?
Yeah, Sean. Oh, go ahead.
No, please go ahead.
This is Dave Galloway. Bill, we had some questions regarding how you assign workplaces and schools. I know you said that things have changed, but one of the things that we notice is that, if you're just going by age, and distance to workplaces, it doesn't seem like the numbers match up. It seems like there should be there's more working age females than there are males, yet there's far fewer females that have a workplace assignment. And so we wanted to know what you used to determine that.
Yeah, I remember that email now. I apologize for not replying to it yet. I think probably we should have a call about that, and I can describe how we do the workplace assignments. Because part of it will depend on the version you're looking at, but I think in general, I would say that the workplace assignments are somewhat questionable. So I'm not surprised that there might be some strange stuff in there. But we can talk more about that and see if there's a way to improve that.
Oh, that's perfect. Thank you.
But Bill, I still imagine that the women that are assigned to workplaces are making 70% less than the men in your synthetic population.
Yes, that's what the data says. I don't know. We don't look at that. But one of the things we don't do in the workplace assignments is try to match based on occupation or industry. The data just is nowhere near good enough to support that. And it's not just the synthetic population data; it's the business data that we use. To try to assign people to.
Okay. Any other questions for Bill?
Yeah, Sean, I have one quick one. I was really interested to hear about FPOP and the potential to relax limitations on modeling over longer time horizons. I was wondering if you could briefly describe how these population projections are made available, something of the practical, what did the data look like? For instance, in the form of yearly updated populations or some periodic log of changes that happen on some frequency, and are the individuals persistent? I mean, you have a unique ID that you can follow over time.
Right. Yes, we do follow individuals over time. So in other words, we don't, at each time step, we don't just take, we don't just generate a new set of data. We do have the ability to track individuals over time, but only by taking a snapshot at each time step and exporting the results. So in other words, it's all happening in memory as it's running, and the only way to get at those time step results is to export it at the end of each time step. But then we'll have to double check, but I'm pretty sure this is the case, is that you will have the same person with the same ID represented at each time step, unless they die and then they disappear. And if we generate new persons, they get a new ID that indicates they're a new person in the simulation.
Okay, thank you. And so these snapshots, I assume that it's configurable what duration of time you want to allow to elapse in between snapshots. For instance, you could do monthly or yearly or.
Yes.
Okay.
That's true. Yep. And we don't have any FPOP results sitting on the shelf that we've ever doing the whole U.S. would be a pretty big job, and so we haven't done that. It'd be great to get some funding for that. And the FPOP model itself hasn't been released publicly, so that's another, just to lay that out there. And so, you know, we'd have to talk further to figure out a way to make that more open to others to use.
That would be great. We'd be very interested in
okay, well, shoot me an email and we can talk further about it.
Yeah, thank you, Bill.
Sure.
Great. Any other, oh, go ahead. Did you have any other questions, Laurette?
Yes, one, and I will ask you, Sean, to help me ask the question.
Sure.
As you know, we have started at the time that Sean was still at Supercomputing Center to use the Canadian synthetic population to start kind of zooming on Montreal, for which we have a very, very rich characterization of various dimensions of the environment, but we have also two very well characterized cohort on what we call the brain to society pathways, let's say, including a bird cohort for which we have various in fact, we have the whole genome sequencing. We have various aspects of the socio and the cognitive development and so on. And at the other hand, we have an aging cohort that we also have started kind of looking at the evolution of emergence of disease and things like that, and within the same environment. So the question we were discussing this week is, to what extent could we look at projecting for what we know of our bird cohort to see what's happening over time, but then use also our aging cohort to kind of start to get at the life course a real understanding of what happened with repeated exposure, with accumulating layers of environment, but also understanding of the biological factor that influences. So, Sean, I don't know if you can help addressing my question to Bill within the context of, you were talking about the ability to interpolate. So, Sean, if you can refine my question to pick Bill's brain as effectively as possible as we are having this online discussion.
Sure. Yeah. So, Bill, the SPEW team at CMU made a synthetic population, or what they call synthetic ecosystem, of the whole of Canada, and we extracting Montreal out of there. So Laurette has some data, this Maven study, that is a birth cohort that they've been following, that we would like to first off be able to take the characteristics, some of the things that you have been talking about, that's enriching with additional behaviors and characteristics, extrapolating that cohort to the larger population, but then also understanding how some of those characteristics may evolve over time as the agents age and move forward in time. Some of those would be easy, like genetics, for instance, doesn't really change that significantly, so you could just kind of assume if that person had this gene at age 5, they have this gene at age 50. That's easy. But some other things might be a little bit more difficult, and would, as you were alluding to earlier, that process would be, it's a forecast, so it has some error codes on it. And Laurette also has a later population that she was, I think, and correct me if I'm wrong, Laurette, would like to use to maybe help correct that or help to understand what. No. Yes? No?
Yes, exactly.
Oh, okay, you said no. That's why I was.
No. Yes, and for this aging population also, we have all type of other information. For instance, for a given status, we have permission to look at healthcare utilization and things. So it's kind of the question, to what extent could we look in some of a nested manner in some way in building this more complex understanding?
So I guess essentially using two different time slices of population with different characteristics to create one super population that ages over time.
Okay.
Put it into vernacular.
Yeah. So, all right. So I'm trying to get my head around this. So let's say you've got some genes, you know, in this child birth cohort, and you want to project how. Well, can you give me like one characteristic or one aspect that you want to try to project into the future?
Okay. Yes, in the sense that the genes that we are interested in are those that modulate the responsiveness of an individual to the environment. So here, it would be about looking at various eating, physical activity, and other characteristics in relation also to the, and then kind of look at what comes later from that. Does it help understand?
Yes, I think so, but it's not something I know much about. I guess if you've got a birth cohort where you know certain things about their, you know, the how a particular gene is going to affect their susceptibility, let's say, or their let's take physical activity, and you have an aged cohort and an elderly cohort where you know perhaps their history of exercise or physical activity as well as their current status. Okay.
Yeah.
Well, I think that would. It sounds like I'm passing the buck here, but I think that would primarily be kind of a statistical analysis to create possible trajectories, and you aren't going to have the same trajectory for every person. There'd be like you'd have like an expanding. You know, take two lines in a graph, and they expand and get wider as you go out in time. And between those two lines, you have possible trajectories of physical activity, let's say, over time. So I think it seems to me like what you could possibly do with the data sets you have is calculate the bounds of those trajectories, and then when you run the model, you take an individual who has, let's say, a characteristic gene that says they're more likely to exercise, or such a thing exists or not, but and then you could say, well, here are the set of likely outcomes based on what we know of an elderly population, and then you'd have to run the model many times to get many different results and kind of average them together to see if you're coming close to getting the right characteristics of the synthetically aged population.
Thank you. It helps. So I will. It's already 12:30. So I'm letting you guys to close, Jay and Sean.
Okay. Yes. Great. Well, again. Thank you, Bill, for an incredibly wonderful talk. It's always good to have you.
Thanks very much.
We really appreciate it. And we'll just tease the next bridge webinar. Will be tentatively Patrick Somerville from Link, which is a company that does static network analysis for a number of different applications. And he's going to come and talk to us about applying network analysis to things like governance and international development. So we look very much forward to it. We haven't set a date for that yet, but we'll be sending out invitations as soon as we do. So thank you very much for joining, and again, thank you, Bill.
Thank you very much.
Thanks for the invitation. All right, my pleasure. Bye. Thanks very much. Bye-bye.
Bye.